My name is Raquel Vasquez, and I'm a Pheo patient. Again, this webinar is produced by the Pheo Alliance, a 501c3 organization whose mission is to empower patients with pheochromocytoma or paraganglioma, their families, and medical professionals through advocacy, education, and a global community of support while helping to advance research that accelerates treatments and cures. A special thanks to Progenics for making this webinar series possible through an educational grant. I'm here with Dr. Lauren Fishbein of University of Colorado Health. Before I provide a proper introduction of Dr. Fishbein, I'd first like to take a moment to provide an agenda for tonight's webinar. Dr. Fishbein will present for about 30 minutes. Then we'll take a Q&A from the community. You can type your questions into the chat section or message us on Facebook. We'll do our best to have all your questions answered if time allows. Many people will ask variations of the same question, so listen carefully for questions that are similar. Let's get started. Again, my name is Raquel Vasquez. I am 31 years old and I am a Pheo para patient. I've been diagnosed with 3 paras. I was diagnosed in November of 2017, and I was diagnosed by Dr. Lauren Fishbein. I had 3 paras, one in my abdomen, next to my abdomen and aorta, and then one on my vagus nerve and one on my carotid tumor. They've all been removed, which has been great. The last one was wrapped around my vagus nerve and provided some difficulty. I lost my voice, I lost the ability to swallow my own food, speaking was inhibited. I've overcome all those things, which has been great, and a long road. Anyone that's dealt with these things can definitely sympathize with those. I've just hit my 2 years of maintenance, and right now I'm free and clear, which has been great. And now I would like to introduce Dr. Lauren Fishbein, who's an assistant professor in medicine at the University of Colorado School of Medicine in the Division of Endocrinology, Metabolism, Diabetes, with a secondary appointment in Division of Biomedical Informatics and Personalized Medicine. Dr. Fishbein's clinical interests are neuroendocrine tumor genetics, with a special focus on pheochromocytomas and paragangliomas. She serves on many communities focusing on para and Pheos, including the Pheo Alliance Medical Advisory Board. I will now turn it over to Dr. Fishbein to present.
Thank you so much, Raquel, and thanks for being here today. And thank you to the Pheo Alliance and Progenics for making this webinar possible. I'm excited to be here today to hopefully teach a little bit about Pheo genetics. I have nothing to disclose. The outline of the talk today, we're going to start with a genetics lesson. I think a lot of people on the webinar probably have a different level of knowledge about genetics, so we're going to start with the very basics. We're going to get up to some complicated stuff, so if you don't understand everything, that's okay. We're then going to talk about genetic testing results, as well as then the inherited syndromes associated with Pheo and the risk of tumors with each of those syndromes. So we're going to start today by talking about personalized precision medicine. So what does that really mean? Well, we're all unique individuals. Just like our thumbprint is unique and defines us each as individuals, so does our DNA. And DNA has some variation in it. Obviously, we're more similar in our DNA. That's what makes us all humans. But we do have some variation that separates me from you. So personalized medicine, my interest began in this in the mid-1990s when I read an article about the promise of the Human Genome Project. And this article talked about how we would use DNA one day to prescribe different medications to a person and also to assess their risk for different diseases. And to me, that was fascinating and what drove me to pursue becoming a physician scientist. And I remember the day when I was in grad school. I was about in the middle of grad school when the Human Genome Project was published. I can remember where I was when these articles in Nature and Science came out. It was a very exciting day for someone who's a genetics geek like me. So let's talk about personalized medicine. What we mean when we talk about this is that healthcare is evolving from a one-size-fits-all to a system that provides predictive, preventative, and precision care. So if we have all individuals in a bucket, the traditional way of treatment or of screening is to screen everyone the same way. And that works for most people, but there's a small subset of people that may not work for as well. And instead, if we do precision medicine, we take a tailored approach where we define people by their genetics, or we define people by a certain characteristic of their disease, where we can group them and give them the best treatment and the best screening based on their genetics and their personal condition. To do this, especially from a genetic standpoint, takes a lot of people. It takes people who know about biology, people who know about medicine, people who know mathematics, computer science, and bioinformatics. We need the technicians. We need the genetic counselors. We also need the ethicists. So why do I bring in ethics? Well, maybe some of you saw the movie Gattaca from the mid-1990s. Great movie, Ethan Hawke, personal favorite of mine when I was younger. Well, this movie is about how people were defined by their genetics, and the government took away people's rights based on your genetics. And so because of that fear, although that was Hollywood, of course, there was still this fear that that could happen in real life. And in 2008, a law was published called the GINA Law, or Genetic Information Non-Discrimination Act. And this act protects Americans against discrimination based on your genetics. So healthcare, insurance, and as well as your employment, your jobs, cannot discriminate based on genetic testing. Now, this law, of course, does not, unfortunately, apply to life insurance, so that is important to know. So what are the pros and cons of personalized precision medicine? Well, the pros are that it really has already and will continue to transform healthcare with new diagnostic and prognostic tools. It will increase our ability to provide the right drug therapy to get the right outcomes for an individual patient, as well as identify biomarkers for the same, for targeted therapeutic treatment. And the possibility exists to improve healthcare outcomes and make healthcare more cost-effective overall. Now, of course, the cons are that it's more money up front to have genetic testing for everyone, and we have to address these public policy issues like healthcare payers and privacy issues with the medical records. But I think the pros and the benefits of this really do outweigh those cons. So our ultimate goals are to provide personalized medicine in preventing or treating complex diseases. We want to provide knowledge for disease risk, and we want to provide individual drug therapy when we talk about personalized medicine. For this talk, we're really focused on providing knowledge about disease risk based on genetics. So before we can get into pheoparagenetics, we want to understand what is DNA. Now, many of you may already know this, but some may not. So DNA is a genetic code determining characteristics of a living thing. DNA looks like a twisted ladder in our cells, and each rung of the ladder is made up of 4 types of blocks, or bases we call them, and these bases pair with each other. We abbreviate the bases by calling them A, T, C, and G, and there are 3 billion pairs of these bases in the human genome. And the sequence of these blocks or letters matter because they will give the instructions for how to create proteins that make our body work. So let's dive into that a little bit further. When we think about DNA, it really is just a sequence of these 4 bases, A, T, C, and G. But those bases will break up into 3 little words, or codons as we call them, and these 3 words go together to build sentences that could be the genes. And when we talk about genes, we're talking about the part of DNA that tells the cell how to make a specific protein. And in order to do that, the DNA has to be transcribed into a message, and that message is called RNA. And that RNA is then giving the instructions to build a protein. And we have tens of thousands of proteins in our body that we need to build the human body and have our cells and our body be functional. So let's talk a little bit about understanding variation in DNA. When we think about variation in DNA, we all have them. That's what makes us individuals and makes each human different from another. We have slight variations in our DNA sequence, and most of the time it has little impact or no impact on our health. So the way I like to think about that is to think about the American versus the British way of spelling words. So theater with an ER or an RE, or labor where you add the U or you don't add the U. Those words mean the same thing as each other, but they're slight variations in how they're spelled. Those are slight variations in DNA, for example, but it doesn't affect the meaning of the word. On the other hand, we can have variations in our DNA that do affect the meaning of the gene or protein or word, as we'll call it. And those examples could be where you take the word state and you convert a T to an L for slate, or you take eat and you insert an S in front, it becomes seat. These are one single letter variation, but it drastically changes the meaning of the word. Those are variants in DNA that are damaging. So when we think about variants that don't change the meaning, those are benign variants. We all have them. When we think about variants that change the meaning, those are pathogenic variants or mutations. Okay, so if you go and you can get clinical genetic testing results, the results can come back and say, no change was found in the genes we tested. That's great. But it can also come back with some other classifications that I wanted to review now. Can come back with a classification saying we tested these genes and we found a benign or a likely benign variant. Now remember, that's like theater with an ER and RE. It doesn't change the meaning. These variants don't matter. Yes, you have them, but it doesn't make a difference. They're not disease causing. They're not associated with any disease. The results could come back and they could say there's a variant of undetermined significance, or VUS we call it. Those variants are usually benign, but we don't really know that for sure. The research hasn't been done to determine if that change in the DNA is disease causing or not. So we call it a variant of undetermined significance. In this case, I will let you know that most of the time, if more research is done, most of the variants of undetermined significance are later considered benign. Now, there are exceptions to that rule, but in the vast majority of cases, a VUS is usually, if it's reclassified, it becomes benign. The third group of classifications is that of likely pathogenic or pathogenic. And remember, these are the ones like eat to seat, where the whole meaning of the word changes because of a variant in the DNA. These are the variants that we know from a research and a clinical standpoint affect the protein function and can be disease causing. These are the ones that we're most concerned about. So if someone is sent for clinical genetic testing, which all patients with Pheo should be, then they can test a number of different ways. The first way is by doing panel testing. This is probably the most common thing that's done for patients who have Pheo. And this panel will cover the 12 most common genes. Sometimes that panel will have a few more genes. And if you're talking to the genetic counselor and it turns out that your family has a very strong history of other cancers, they might decide to do a very large panel of genes. It could be 84 genes, it could be 100 genes. It all depends how many genes they test based on your personal history and your family history. Sometimes we'll just test one gene at a time, and that can be the case in certain conditions. If someone has a very clear clinical syndrome that clinically we know they must have a mutation in this one gene, and an example would be someone who has multiple endocrine neoplasia type 2, MEN2, or VHL, it might be very obvious clinically, and we might not do a full panel. We might just do the gene that we're at the highest level of suspicion for. The last type of testing I want to mention is specific variant testing. This could be the case when a family member had a Pheo. They may have gotten a full panel testing and they found a pathogenic variant in one of the genes that leads to Pheo. Then the rest of the family will just be tested for that specific variant. And in that case, we don't test the whole gene, we don't test a panel of genes. We literally just look for the one change in DNA that we know is already in the family. And that's called specific variant testing. So when the results come back, it may say negative. And what that means is that there was no pathogenic or likely pathogenic variant found, no mutation found. It might be that there are still a VUS, a variant of undetermined significance, or a benign change that was found, but there was nothing found that we think is leading to the disease. Now, it's important to know which genes were tested, and the report that comes back will actually list, was it those 12 genes? Was it 84 genes? Did they only test 1 or 2 genes? That's important to know, especially when the result is negative. We can also get positive results. When we say positive results, we mean we found a cause of the disease, a pathogenic or likely pathogenic variant in one of the genes that was tested. And then again, we can have those VUSs, those variants of undetermined significance, where we're not quite sure if it's disease causing or not. When the results come back and it is positive for a pathogenic or likely pathogenic variant or mutation, you're going to get a couple bits of information about that specific variant. Now this is high-level information, but I included these slides based on some of the questions that were pre-sent in for how to read the positive results. So when the results come back, you're going to get a gene name, a position in the DNA with what the change in that base pair or letter was. You're going to get the position in the protein and what that amino acid or word change was, as well as the classification. So let's look at an example. Here's an example of an SDHB gene that has a change from a G to an A at the DNA position at 137, and then a change in the protein at position 46 from an arginine to a glutamine amino acid. Now you don't need to know these details, but if you're interested in what the variant your provider is looking at, this is the information we're looking at. What specific change in the DNA and the protein did they find when they did the DNA testing? So another question came in about, do companies retest for more genes as they become known? And I wanted to address that here. The answer is no. Companies don't keep your DNA and retest it in 5 or 10 years if a new gene has been discovered. So if your original genetic testing was negative, I always suggest to discuss with your provider in 5 or 10 years, research moves a little bit slowly, if any new genes have been discovered and if you should be retested at any point if you are negative at the start. However, if a variant is reclassified, so meaning a VUS got changed to, let's say, likely pathogenic, that's very rare that that happens, but if it did happen, the company does in fact contact the patient as well as the provider who ordered the test to make sure that people are aware that that variant has changed classification. Again, that's exceedingly rare that that happens, but the companies will reach out in those cases. So let's take a minute and talk about the inherited genetic syndromes associated with Pheo-para. So Pheos and paras are neuroendocrine tumors of the adrenal medulla and extraadrenal ganglia. So what do I mean by that jumble of words? Well, pheochromocytomas are the tumors that form inside the adrenal medulla. The adrenal glands sit on top of the kidneys, and if a tumor forms inside the adrenal gland, we call that a pheochromocytoma. Now, tumors can form in any of these other nerve bundles or ganglia from the skull base all the way to the pelvis. Those are called paragangliomas, or paras for short. And sometimes we separate out the head and neck paraganglioma because they usually are not secreting adrenaline hormone like the rest of the tumors. So sometimes we separate those out. Now, of all pheochromocytomas and paragangliomas, 30 to 35 to 40% of these are associated with hereditary syndromes. And we're going to talk about each of these syndromes and what else can be associated, and also what is the risk of developing a Pheo-para if you have one of these syndromes. So as I mentioned, 30, 35, 40% of these tumors are hereditary, and in kids that number is even higher. About 80% of Pheos and paras are associated with a hereditary syndrome in children. And we now have over 12 susceptibility genes, meaning genes that we know, if they're mutated, increase risk for developing Pheo-para. You can see here on this slide that in the 1990s, we only knew of 2 or 3 different genes that increase risk for Pheo-para. But as the decades have progressed, we are now up to 12 genes. There are a few more that have been discovered in the last couple years, but they're not yet proven without a doubt to be associated with Pheo-para. So these are the 12 most common that we feel pretty strongly have good evidence to show they're associated with Pheo-para. So let's review these syndromes. The first is neurofibromatosis type 1. It's caused by mutations or pathogenic variants in the NF1 gene. Neurofibromatosis type 1 occurs in about 1 in 3,000 individuals, and these individuals have a variety of different conditions associated. One is to have a number of cafe au lait spots, birthmarks, but they have to have at least 6 of them and of a certain size. neurofibromas, which are peripheral nerve tumors, and a number of other things you can see on the screen. Now, it turns out pheochromocytoma is not part of the diagnostic criteria for NF1, but people that have NF1 are at a higher risk for developing pheochromocytoma, about 1 to 13%. They're also at risk for other neuroendocrine tumors, a pancreatic neuroendocrine tumor, slightly higher than the general population. The second condition we're going to talk about is multiple endocrine neoplasia type 2, or MEN2. This is caused by variants in the RET gene. It occurs in about 1 in 40,000 individuals, and patients can have MEN2A or MEN2B. They're very similar but slightly different conditions. Both conditions are associated with pheochromocytoma. About 50% of patients with MEN2 will develop a pheochromocytoma. The graphic shows MEN2A and B. It also shows MEN1. Now, MEN1 is not typically associated with Pheo. It can occur, but exceedingly rarely, and those patients develop a different set of tumors associated with endocrine conditions. The third syndrome I want to talk about associated with Pheo-Para is von Hippel-Lindau disease, or VHL. VHL is caused by mutations in the VHL gene, occurs in about 1 in 36,000 individuals, and individuals who have this syndrome are at risk for developing a number of different tumors, from pancreatic neuroendocrine tumors to kidney cancers to pheochromocytomas. About 20% of individuals with VHL will develop pheochromocytomas. The next set of conditions I want to talk about are the ones that were discovered just in the 2000s. These are the hereditary paraganglioma pheochromocytoma syndromes. They're caused by mutations in the succinate dehydrogenase gene complex. That's a mouthful, I know. We call it SDHX, or the SDH genes. There's A, B, C, D, and a gene called AF2. And mutations or variants in any of these genes can increase risk for pheochromocytoma or paraganglioma. Patients who have one of these genes, they may be at risk for developing Pheo-Para at different locations in the body. They're also at risk for kidney cancer, as well as GI stromal tumor. Now, the risk of those last two are a little bit smaller than the risk for Pheo-Para, but they're still present. The last thing I want to mention about this group of syndromes here is that SDHD, D for dad, is inherited through the paternal side. So what that means is that if you develop the gene mutation from mom, then you are less likely to develop a Pheo-Para. But if you develop the gene mutation from dad, you're more likely to develop Pheo-Para. It's a complicated thing that I'm happy to take more questions about. So those are the most common genes associated with Pheo-Para, but there are some more. This table lists some of them. I don't have time in this webinar to go through every single one of them, but I wanted to at least list them here so that people have heard of them. And if you have any questions, I'm happy to discuss them further as well. So should a person be tested if they have a Pheo-Para for any of these syndromes? And I would answer a strong, resounding yes. And the reason is that 35 to 40% of Pheo-Paras are associated with an inherited germline genetic pathogenic variant or mutation. And that's a high percentage. So we do recommend that everyone get tested. But the question is why? What difference is it going to make? Well, it's going to make a difference because it's going to allow us to screen that person properly for the additional Pheo-Paras that might be associated, for any other cancers or syndromes that are associated, and also it helps determine the family member's risk for developing any disease. Will every person with an inherited gene mutation develop a Pheo-Para or other feature of these syndromes? I get asked that a lot. And the answer is no. This is not extremely common or penetrant. So let's talk about what that means. When I say not everyone's going to develop disease, that's because the penetrance is lower. So what does penetrance mean? Penetrance means what is the risk of disease. Expressivity is also important, and that means what type of disease is the person going to get. So let's talk about this in a little more detail. Again, these next couple slides are a little bit on the more advanced side, so don't worry if you've never heard this before and it's a little complicated. We can go through more details. So when I say penetrance, I'm talking about risk of disease. So if each of these little squares is a person, and green is a person with disease, and white is a person without disease, you can see that we could have complete penetrance, where every person who has the gene mutation is going to develop disease. Or we can have incomplete penetrance, where not everybody is going to develop disease. Only a certain percent of people with the gene mutation will develop disease. Then we talk about expressivity. And this is where someone may have a mutation in a certain gene, but all their family members who have that mutation get a different set of symptoms. One person may have a neck paraganglioma, another person may have an adrenal. The expression of the disease is different, and that's what expressivity. Narrow expressivity means everyone gets the exact same thing. That rarely happens. Variable expressivity means each person gets a slightly different type of disease. So when we talk about pheoparagenes, we're talking about incomplete penetrance. Not every person with a gene mutation will develop a pheopara or another syndrome, another feature of the syndrome. When we talk about pheoparagenes, we also talk about variable expressivity. So the types and locations of tumor that a person gets will vary. Okay, so let's summarize the penetrance. This was one of the questions that came up. And when we talk about what's the risk of developing a pheopara, we're usually talking about people who have the gene change, the gene mutation, but have never developed disease before. That's an unaffected carrier. And these people may have been found because their family member was tested and then they got tested, or they had a different cancer, had a large set of genes, and discovered they had a pheopara gene. And in those cases of unaffected carriers, you can see here the penetrance of disease. The highest penetrance is in RET and SDHD. That means if you carry an SDHG mutation, for example, you have about a 43% chance of developing a pheopara by age 60. In contrast, people with an SDHA mutation, they have very low penetrance. By age 70, it's only 10% chance of developing a pheopara. That's very different depending on the gene. So how do we screen mutation carriers? Well, for NF1, only 1 to 13% of patients will develop a Pheo. It's usually in the adrenal gland and it's usually secreting. So the guidelines for NF1 say that actually only NF1 patients who have high blood pressure or hypertension should be screened for Pheo. And we screen with the plasma metanephrines or 24-hour urine metanephrines and catecholamines. For MEN2, about 50%, it's a high risk. But they're almost always adrenal Pheos and they're almost always secreting. So the guidelines say that basically everyone with MEN2 should be screened once a year with plasma metanephrines or the 24-hour urine tests. For VHL, the risk is about 20%. Again, usually these tumors are adrenal-based Pheos and usually secreting. And again, the guidelines recommend that everyone with VHL be screened once a year at least with plasma metanephrines or 24-hour urine metanephrines and catecholamines. For the SDHX or FH gene mutations, these are a little more complicated because, as I showed you, each gene has a different risk of developing a tumor. Each gene has a different risk of developing secreting or non-secreting tumors, and the tumors can be in different locations. So the guidelines here recommend that everybody with these gene mutations get screened at least once a year with those plasma metanephrines or 24-hour urine tests. But in addition to that, we recommend imaging every 2 years from the skull base to the pelvis. And that's to pick up any of the non-secreting tumors which can occur in this particular group of people. So one of the questions turned in ahead of time was, well, what about sporadic tumors? What if I don't have a mutation? How did I get a Pheo or para? Well, that's a good question, and we don't totally know the answer, but it's important to think about tumor genetics versus the inherited genetics that we've been talking about. So when I talk about inherited genetics, which we've discussed this entire webinar so far, we're talking about mutations that occur in what we call the germline. That means you're born with it. Every cell in your body has this change, and we usually test that in blood or saliva, and it's to see what the predisposition for developing tumors is. But we can also talk about genetics of the tumor itself. And what happens here when we talk about these tumor genetics, or somatic genetics as we call it, we're looking for a DNA change that happened in one cell in your body, not your whole body, but one cell. And that cell grew and proliferated into a tumor. And we can test for just mutations in the tumor. We don't do that very often, but in patients who have metastatic disease, we might try to do that to see if there's a therapy that matches that DNA change. So the patients who have sporadic disease, non-inherited Pheos or paras, it may be that some of these same genes got turned on just in the tumor, but not in the whole body. If that's the case, there's no risk for passing this on to family members. There's no risk for other family members, or no increased risk for other family members developing disease. So I just want to end by summarizing what we discussed today. DNA variants, they can be benign or pathogenic. Many inherited genetic syndromes exist that predispose to Pheopara. And not everyone with a Pheopara will have an inherited syndrome. And testing is really important, in my opinion, because it helps us understand the risk to that person based on their additional risk for Pheoparas, as well as other disease features. And it helps us understand their family members' risk for developing disease as well. So I want to thank you for your time, and I look forward to answering any questions that you might have.
Perfect, thank you so much. We'll go ahead and start with the Q&A. Our first question is actually interesting. I'm assuming it's a SDHB question, which is what I have, which is very interesting. What is the best age to test to see if my daughter has a genetic mutation her father has?
Sure, so the answer varies depending on a lot of things. So what's the best age to test a child? The guidelines do have their opinion on what the best age is, and that depends on the syndrome. So if it was SDHD, for example, the guidelines for the SDH genes recommend that we test kids around ages 6 to 8 years old. But there is a lot of parents that I have discussion with who feel like that's a little too young to start testing. Other people test as soon as a child is born because they want to know. So I think it's really important to have a conversation between the parents, sometimes the child if the child's already a teenager, and the provider to determine what the best time is. If the genetic testing isn't done early at the guideline recommended age, then I usually recommend that we at least take blood pressure, we at least look closely for any signs or symptoms that might suggest disease. But each gene has a different age that's recommended, so it really depends on the gene. It's hard to answer specifically.
Okay. My daughter has had a Pheo removed and is SDHB, but no one in the family has it. How is this possible?
So I assume that this person is asking that no one in the family has the SDHB change, as opposed to no one has the Pheo. But let's start with no one has the Pheo. So if the daughter has the Pheo and has SDHB, it's very possible, due to that penetrance, that risk of disease for SDHB is only about 25% by age 60. So it's very possible for no one else in the family to develop a Pheopara, but they may have SDHB mutation. Now, if you're asking about why the mutation isn't seen in anyone else, that's a very complicated issue. One possible reason is that as the egg and sperm joined and started development, there was a mutation that was caused, and in SDHB, we call that somatic mosaic. That's a much more complicated topic than I think we can discuss on this webinar today, but it's basically a change in the DNA that occurs after conception instead of before.
Okay. Who pays for genetic testing?
Very good question. So in the past, this was a big concern for who's going to pay for this genetic testing, and it used to cost a lot of money. Well, the costs have come down as technology has increased for genetic testing, and insurance almost always covers genetic testing. So I think I've only had a handful of cases where that's not true. Now, the cost out of pocket can vary, and so genetic counselors are great at sort of helping guide how much is this going to cost before they actually send the test, so that you can make the decision if it's appropriate and affordable for you to do the testing.
Can you talk a little bit about SDHAF2?
Sure. So SDHAF2 is one of the SDH genes, part of that whole syndrome that causes Pheopara. SDHAF2 is one of the newer genes that's been discovered. We don't know a lot about it. It probably is found in only a small percent, maybe 2% or less of patients who have pheopara. It mostly is associated with head and neck paraganglioma, and it's usually bilateral disease. But again, it's one of the more rare genes, so more research needs to be done to really define the characteristics, but it's usually bilateral neck paraganglioma.
That's interesting. How does diabetes affect pheopara?
So how does diabetes affect pheopara? I would actually ask the question the other way. And the reason I would do that is that pheopara can secrete those adrenaline hormones. And those adrenaline hormones can cause high blood sugars. So people who have a secreting pheo or para tumor, their diabetes, they may not have had diabetes before and now they do, or their diabetes can worsen with a secreting tumor. How diabetes affects pheopara, I don't think we really know that yet. There's a little bit of evidence that both diabetes and pheopara can affect bone health, but this is very new research that's ongoing. So hopefully we'll learn more with time.
Should I avoid coffee? No. Personally.
Well, listen to Raquel. So yes, I agree. I think if there is someone who has secreting pheopara and high levels that can't be controlled, caffeine can increase the symptoms. So the symptoms of high adrenaline, which was not really covered in the webinar today, are palpitations, rapid heart rate, sweating, things like that. And so caffeine and coffee can exacerbate that in someone who has secreting disease. But otherwise, there's no need to avoid it.
How do I find a doctor that has experience in pheoparas?
Very good question. So I would use your resources. The Pheopara Alliance is one of them. They do have a doctor tracker that is being worked on every day to increase the number of doctors and providers, especially in the US, that we can find that know a lot about Pheopara. The Neuroendocrine Tumor Research Foundation has a similar one as well. So reach out to these organizations because they're here to help you gain information, gain knowledge, and find providers who know about the disease.
Are there any updates on research regarding turning gene mutations off?
So I'm not exactly sure what this person means about turning a gene mutation off. There's always research about gene therapy in general, about trying to replace genes that don't function or turn off genes that are over-functioning. In Pheopara, we don't have a lot of results in this area at this time, unfortunately.
What is the chance of recurrence for a sporadic pheopara?
A chance of recurrence for a sporadic pheopara. This is a good question, and there's a lot that goes into that. So a sporadic pheopara that's removed in its entirety probably has a low chance of coming back, but I still recommend that every person who's ever had a pheopara get tested at least once a year because we don't know if it's going to return. If we lump all pheoparas together, sporadic and the hereditary, there's about maybe a 25% chance of recurrence or widespread metastatic disease at some point in the lifetime. So we want people to always be checked at least once a year with the blood tests or urine tests to make sure that we catch any recurrence early.
Okay. Speaking of the urine test, can a pheopara be missed on a 24-hour urine collection test?
So I did mention that some of these paraganglioma are not secreting. So if the tumor is not making adrenaline hormone, then we won't pick it up on the urine test or the blood test. And that's why in some cases imaging is important. But remember, if the tumor is not secreting, then it's probably not causing symptoms of the high adrenaline because it has to be making the high adrenaline to cause those symptoms. Sometimes the non-secreting tumors can cause symptoms because there's pushing on other things, especially in the neck. They can push on the nerves and the vessels there. But they're not usually causing symptoms if they're not the symptoms of the high secretion. We can miss the non-secreting tumors.
We're in Colorado, so the obvious question came up. Can medical marijuana alleviate pheo or para symptoms or cure them?
So very good question, and the simple answer is we have no idea. There's been no research done. I will tell you that when I'm testing someone for Pheo-Para, I have seen that people who use marijuana, they can get false positive elevations on their lab tests. So in the testing period, I would be cautious about using the medication.
After an adrenal gland is removed, is it still possible to have fight or flight symptoms?
So if an adrenal Pheo chromocytoma is removed, then sometimes it takes a while for people's systems to get back to normal. If your tumor was making lots and lots of hormone and all of a sudden that's removed, it takes a while for the body to kind of reach equilibrium again. But certainly those symptoms should decrease pretty quickly, and in a lot of patients they go away immediately after surgery.
Can you have a Pheo on one adrenal gland and then have it removed and then one on the other, I'm assuming? So have one Pheo on one gland, have it removed, and then have one come on another?
Yes, absolutely, especially in these genetic conditions with multiple endocrine neoplasia type 2, VHL in particular. They're very prone to having a Pheo on one gland and one develop on another, and that can either appear at the same time or they can happen years apart. So again, really important to always be tested at least once a year for the adrenaline hormones to make sure we catch that early.
Is it unusual for someone to have a GIST or para in a kidney or liver but no other abdominal locations?
So a GIST and a para are two separate tumors. So a GIST is a GI stromal tumor. They occur within the GI tract, and in the SDH mutations, they're usually in the stomach, but they can occur anywhere in the GI tract. So that's a different tumor than a para. And a para or a GIST can happen anywhere in the GI tract, and a paraganglioma can happen anywhere in the body from the skull base to the pelvis. So it's not unusual to have them in sort of odd locations or outside the adrenal gland, for example.
I have an SDHB GIST. I had a uptake on the gallium-68. Could my tumors be partially neuroendocrine tumors?
So this is a complicated question where a lot of research is going into. So the data that have been out there, we don't have much about looking at the GIST tumors using gallium-68 scans. But some of the data that's been published has shown that most GISTs don't take up activity on the gallium-68 scan, but sometimes they do. And so to answer the question whether there are actually neuroendocrine cells inside that GIST or not is a hard one, because we haven't then taken them out and looked under the microscope. But what it tells us is that gallium-68 scans are really picking up a certain receptor that's usually found on neuroendocrine cells. But that receptor might be being misexpressed on a different cell, and so that's why it lights up the GIST. Or maybe there is a neuroendocrine component, although from what we know about GIST, that would be rare. So unfortunately, don't know the answer to that question, but people are researching it.
Very good. Can you talk about some of the radiation therapies available?
Okay, so I'm not sure what this person means by radiation therapies. I'm going to talk about a couple things. So there's radiation therapy that we would give to lots of different cancers that are external beam radiation, where we shoot radiation into the body to focus at a specific point in the body where there's tumors. That's a classic term for when we say radiation. We'll see a radiation oncologist for that and irradiate specifically maybe a metastasis to our bone, or sometimes even lymph nodes inside the body and things like that. We also use radiation a lot for head and neck tumors that cannot be removed by surgery. They may also mean radiation by nuclear medicine treatments. So the one that's currently FDA approved is called a Zedra. It's MIBG. It's not really radiation, but it uses that a little bit. It uses a radioactive molecule that we inject in the body that targets the tumor and then gives off radiation inside of us to kill off that tumor. And so there is a therapy FDA approved called a Zedra, that's MIBG, to treat metastatic metastatic pheopara or unresectable pheopara. There's another that's being studied in clinical trials called Lutathera, or PRT. That's another nuclear medicine type of radiation. That therapy has been approved for the GI neuroendocrine tumors, but it's still under clinical trial for pheopara.
Okay. What is the survival rate for someone that has Pheos that has a mets to the liver?
So another complicated question, which is hard to answer without knowing any personal information, other medical history, and the original pathology of the tumor. So it's not really something I can answer. But what I can speak to in general is that people who have metastatic pheopara, some people unfortunately succumb to the disease within a few years, and others can live 20 years or more with metastatic disease. And part of the research that I do in my lab and others are doing in their lab is trying to understand, how do we predict that? How do we know which course of the disease is going to happen? And so far we have maybe some clues still in the research state, and so hopefully we'll be able to tease that out.
If I can ask, when you're talking about the lab, I think it's really important. I've donated all my 3 tumors. I had 2 surgeries in full, so they bought 2, got 1 free from me. Can you talk a little bit about who donating tumors and their companies or the research that goes into them?
Sure. So in rare diseases in particular, I think it's really important to participate in research if you're comfortable with that. And one way to do that is to sign up to donate tumor tissue at the time of surgery. And there are lots of different ways to do that. Some institutions that focus on pheopara research will have protocols where you can consent to donate your tissue to the researcher at that institution. And we take it to the laboratory and we try to understand why the tumor formed. Can we identify markers that might say it's more aggressive or less aggressive, or even targets for potential therapies? And we also try to make models. So one of the challenges in pheopara is we don't have a lot of cell culture or animal models to study this disease. And so a lot of us are working to try and do that. And one place in particular that's doing that is the Broad Institute in Massachusetts. And you can sign up to have your tumors, no matter where you are in the US, donated and sent over to the Broad Institute for research to try and develop cell culture and animal models for this disease as well.
Awesome. Can you talk about the frequency of scans and when is it too many? You and I just talked about this.
Yes. So it's a very good question that comes up a lot. So how often should I be scanning? It really depends on the disease state of that particular person. So what I would say is that someone who has a gene mutation but never developed disease, there are guidelines for how often we should screen. And for some mutations and some genes, we never do imaging screening. We only do the blood tests. In others, we do imaging screening maybe every 2 years or so. And then in other people who've already had disease, how do we decide how often to do screening? Well, if their tumor was fully resected by surgery and there's no evidence of disease left, then again, it sort of depends on the pathology report and what the tumor looked like under the microscope to make that decision. Sometimes, again, we might never do imaging. We might only do blood tests. We might do imaging every 6 months or once a year. So it's very tailored to the individual. In patients who have metastatic disease, I would say that we usually image somewhere between 3 months and 6 months, depending on how fast their disease is growing. Most of the time, pheopara is pretty slow growing, so I never recommend imaging. I shouldn't say never, because there are always exceptions, but almost never recommend imaging more than every 3 months.
I've had 2 Pheos and F1. Could spinal tumors be paras?
So paraganglioma can occur in the spine. In the setting of NF1, though, it's a little more complicated because patients with NF1 also develop plexiform neurofibromas, and those can form around the spine too. So it's hard to say whether those are paras or plexiform neurofibromas. Sometimes imaging can help determine which is which, and sometimes it takes surgery to determine which is which.
Is blood plasma test better or worse than a 24-hour urine collection? It's easier.
Yes.
It's a lot cleaner.
So as my patient, she knows I prefer the blood test.
Yes.
I prefer the blood test because it is easier than a 24-hour urine collection. A few years ago, the 24-hour urine collection was the gold standard, though. The blood tests weren't as good. But now we have better tests, and so the blood tests and the urine tests have pretty equal sensitivity for picking up high adrenaline levels. There are things that can interfere with the blood test and the urine test, and those are certain medications or over-the-counter supplements. So sometimes I'll do a urine test instead of a blood test if I think there's a lot of interference that could happen, because the blood tests are a little more sensitive to that interference, although the urine can be sensitive to it too. So sometimes I almost always start with the blood test, and then I'll do a urine if I'm not sure about the levels. If they come back indeterminate, sometimes we'll do a urine. Or in patients who have episodic events or episodes that I'm trying to catch, then it's hard to say, oh, at 11 p.m. come in for a blood test. And so in those patients too, I might send them home with the jug to collect urine to start when an episode occurs, so we can see if their levels are high.
Easier to do in the winter because you have to keep it cold.
Yes.
Is it okay to eat pickled foods?
Ooh, I've never heard that question before. I haven't heard any reason not to. But if it upsets you or isn't good for you, or you notice it aggravates something, then I would say avoid it. But I don't know of any particular research on that topic.
Do we have any more questions? I don't have any questions. Any questions from anyone?
We
No?
have some audience here.
We
Anyone
have
have
some
any
people
questions?
in the audience.
Well, one question I get asked is, what kind of imaging is the right imaging to diagnose Pheo-Para? And sometimes people will ask, is a CAT scan the right one, or an MRI, or should I be doing MIBG scans or a dotatate scan? And that also is a very individualized decision based on that particular person and what's going on with that person. So I would say that the vast majority of the time, if we're trying to detect a Pheo or Para, we can usually start with what we call cross-sectional imaging, so either a CAT scan or an MRI. When we're screening people who have a known mutation, we'll usually also start with MRI to decrease the radiation exposure. The nuclear medicine tests, like an MIBG or a dotatate gallium-68 scan, those are really important for detecting small tumors, or if we are worried about metastasis somewhere, then those tests can be really helpful for us. But I almost always start with a CAT scan or MRI. Also, I should say, part of that decision is cost. So the nuclear medicine tests are much more expensive, and so if we can detect something with an MRI or CAT scan, we're saving some costs. If we can't, we absolutely will go to the nuclear medicine test.
They're a lot easier too, I think, on the body to just go in and have the contrast rather than having the
Nuclear medicine.
the nuclear medicine that has to make it glow.
Yes.
So it's a lot easier. How often do tumors uptake on a MIBG?
That's a good question too. So not every Pheo-Para takes up MIBG. It's not very sensitive, so only about 60% of Pheo-Para will take up on an MIBG scan. If it does take up, it's pretty specific, but it's not very sensitive.
If it does take up, does that mean that the treatment will work?
Good question. So we talked about the Azedra therapy, the nuclear medicine treatment for metastatic disease. And the only way we can offer that as a potential therapy is if the tumor does take up MIBG. So we always start with an MIBG scan in those patients. And if the tumor does not take up MIBG, then we don't usually treat with Azedra or MIBG therapy, because the tumor probably won't take up the therapeutic version either. So if the tumor is positive on MIBG, then there's potential to be a candidate for that therapy.
Okay. What is some promising research about Pheos and Paras?
Oh, there's so much to look for on the horizon, which I think is really exciting. There's a lot of people around the world that are working on this. Research is slow, so bear with us, but a lot of things have been discovered. You saw that graph of just the number of genes that have been discovered in the last 10 or 20 years. So that was a huge advance in the field of Pheo-Para. There are now clinical trials being done on a number of different drugs that hopefully will result in therapies for those that have metastatic disease. we're doing research on trying to find markers. So if the tumor is removed, can we study that tumor to decide, is there a marker that might mean you're more or less likely to develop recurrence or metastatic disease? And there's a lot of research directed on those efforts too. We do a lot of sequencing of the tumors, a lot of integrative sequencing, lots of different platforms, and myself and others are involved in international collaborations to try to answer some of these questions. So I think there's a lot of hope and a lot of excitement out there for what's going to come in the next 5 and 10 years.
This is a great question, because I've heard that they like high altitudes. Is it okay to live in a high altitude place if you're more affected by Pheos and Paras?
Yep, good question that I hear all the time, especially living here in Colorado and Denver. So the question is, we don't know. There's a teeny bit of data that suggests, it's not very good data, but there's data that suggests that maybe people who have a gene mutation in the SDH genes might have an increased number of tumors if they live at high altitude, but it's very bad data because they already looked at people who already had a tumor and then compared those at high altitude and low altitude. They didn't take people who never had a tumor and compare them at high and low altitude if it made a difference. And remember, if you have one tumor, you're more likely to get another tumor. So it wasn't very good data, but a lot of us are looking into that and trying to study that question. So we think there might be an association, but I would not say it's strong enough to tell someone to pick up their life and move to a different state.
I thought you were hoping to tell us to move to a beach.
Right.
That'd be great.
But there's plenty of people with Pheo para that's sea level too, so I don't think we have enough data yet to say that.
Last question. When should I look into participating in clinical trials?
Yeah, so clinical trials are really important, and they're important because we don't have curative treatments yet, unfortunately, for people who have metastatic disease. And even people who don't have metastatic disease, who just have individual tumors, we still don't fully understand why they form, why people get more than one of them. And so there are different types of clinical trials. There are clinical registry trials. I have one of those where we collect information in a big database to try and answer some of these questions. We have registry trials for the tumor that we talked about before, where we take the tumor to our labs and study them. And then we have clinical trials in the classic sense of the word, which means trying different drug therapies in different groups of patients. And there are trials around the country right now, and I would say that they should always be top in our mind of potential treatment options, because that's new cutting edge research, and the way we're going to learn is by participating in these clinical trials to try and find the best treatments for the right patients at the right time.
Awesome. Thank you so much, Dr. Fishbein, for your time and dedication and passion to our Pheo para community. This has been very educational. Personally, and I've told my family to watch, which I think helps being a family of SDHD genes. One last thank you to Progenics for providing an educational grant for this webinar series. We hope all of you at home will continue to connect with the Pheo para community. You can reach out to the Pheo Para Alliance on social media at Pheo Para and on our website at pheopara.org. This webinar will be available in a few days on YouTube and on our website. We'd like to thank everyone who attended today, and we are stronger united. Personally, I wanted to thank you for allowing me to do this. When I was diagnosed, there wasn't a huge community that I knew about that's been the experience with Pheo and Para. My sister and I were diagnosed 2 weeks from each other. And so this has been a great opportunity, and it really means a lot. So thank you.
Thank you for doing this too, and thank you.
Yeah, have a great night.